Cool. Thank you so much, Granny. So yeah, I'm really excited to be here today. I think it's really interesting listening to all the engineering IT teams speak about all this beautiful infrastructure they built their scientists and how I'm very appreciative now, which I'm guilty of sometimes. So scientists don't always adhere and use those infrastructures correctly. So today I'm really excited to talk about how as scientists myself, you can kind of maybe encourage those teams to engage and really see the benefits of storing and using different genomics data with Quilt Data practice. Okay, so just to introduce myself, I'm a computational biologist. I went to grad school in Canada, in Toronto, sort of Cancer Center. And then about 2 years ago, I moved here to Cambridge to work as a computational biologist at Celsius Therapeutics. So sort of within grad school and also my work in industry, the main focus of my research has been to use different types of genomics to discover new therapeutic targets, specifically in oncology. So I had to kind of pin down different sort of buckets of different techniques and experiments I work with. I mainly use single-cell RNA sequencing, in addition to full exome and genome sequencing, and a little bit of genome-wide CRISPR screening. But today I want to talk about specifically whole exome sequencing, which is a method that a lot of researchers use to profile the protein coding regions of different genes to help them understand, especially in oncology, where there might be mutations or different things going awry that they can tie to different outcomes to help them understand these. So what's the problem with exome sequencing data? How can we use Quilt to improve the efficiency of our phenotype? So traditionally, at least from my experience, well, exome sequencing data is traditionally inaccessible. So this data is really big, especially if you have lots of samples, you're working in consortium style effort like TCGA or defects. In these pipeline outputs, after you process the raw data off the sequencer, they're often relatively inaccessible and accessible only by command line. Which doesn't really work for all members of the team, especially if you're working across different tools as opposed to the industry. So yeah, to sort of steal a picture of the Quilt team, you have this network of all these different news of your company and like you're a com bio. But first, like myself, you sometimes find yourself in the middle of this web where you're getting all these different queries in your team days across different teams, asking you where different files are, asking you for different plots, and it can kind of, throw off your to-do list, to put it nicely. And it feels a little bit like make work at the end of the day. Things that you don't feel like you should have to be doing if this data which is accessible and organized and easy. Right. So this is just a picture I want to show to kind of give a little bit of taste of who is using this exome data, why they need to see it, sort of what aspects of the pipeline they would be on hand with different datasets. So I think sort of from a production standpoint, exome data can be broken into these annual buckets. So you need to acquire the sample, you process it, and sequence it. You need to push your pipeline to process it, to control on it. And then you need the rest of the company to basically do all the discovery, the interpretation, validation, and also marketing if you get a product at the end of the day. So there's tons of different teams, how they like first sort of that plug into each of these different steps. And it really spans everything from project management, clinical operations, as well as the platform sequencing for data engineering, comp bio, and also this development if they're looking back at some of these results to help them interpret different assets that they need developed from it. So all these different people need to be able to access and use this data, but as I'm sure all of us know from reading today, not all of them want to SSH into a cluster and search around for these different file types that might be storing them relations. So because we've all kind of discussed this morning, one of the solutions to this is Quilt. To generate a Quilt package, to basically stick everything to your processing pipelines, just the raw data and the execution commands, the output logs, pipeline outputs, as well as any downstream analysis, such as computational biologists and lab cheaper notebooks, any simple raw data, and also custom bioinformatics that in those notebooks, as well as analysis tools, put that all into a Quilt package, where every single one of these aspects of your pipeline is now versions. You can DM your collaborators, shareable links where they can go on the Quilt trees themselves, find these different aspects of the pipeline. And as we'll show in a bit with the demo, you can also visualize some of these files directly in the trees. So in combination with the Quilt team, we did a little case study to sort of highlight the power of using Quilt with whole exome sequencing data, where we downloaded raw exome sequencing data for 9 KRAS cancer cell lines. Found the CCLE, Cancer Cell Encyclopedia Project, the Brown Institute initiatives up years ago. We grabbed that FASTQ data from the sequence tree archive, and then we pushed it into the Nextflow NF Coursera pipeline in tumor-only mode, using the NF built plugin. And this pipeline uses this alignment and food processing, even variant calling the annotation and generating a bunch of summary results, including the multi-QC report that Johan highlighted previously. And then we also did a section of downstream analysis that would mirror what computational biology team would like when doing different sort of bespoke analyses to analyze the typical results coming out of the process of pipeline. So what I hope to show in the next couple of slides is how you can use Quilt packages to essentially enable efficient data access across all of these steps, help you track your data and share your data with different teams across all these different stages of the genomics. Okay, so I think Johan did a really great introduction at the end. Clone sequencing, very similar to that. There's an excellent core community called SERIC, which is used to basically go from raw data per sequencer and process different whole genome, exome, or targeted sequencing panels to all the Lyman processing C. Very common, and also spit out lots of base reports to help you visualize the quality of your data. So we ran this pipeline on this CLE public fast queues that we obtained from the SRA with the NF-Quilt plugin, which nicely packaged all the outputs of this pipeline to a nice interface where you can click around and see all the results easily without having to do really any manual intervention, aside from just specifying this plugin when you write your mixed work. So what does this look like as a user? I had a lot of fun actually checking out these different results. You can imagine sort of different questions you would get asked. Update your data back for exit sequencing. At least in my experience, it's always kind of a rush to look at the data and get all the latest results out of it. So some questions that your platform team might want to know is how does this FASTQ look? It looks a little wonky. It's library trace and want to know the quality of the sample. So you can check out the multi-QC report in an interactive way through the Quilt interface. And you'll also get these different links that link directly to that report or a file that you're talking into cloud this. Additionally, different meetings, so one's like, hey, what that bio person actually do behind the scenes to sort of build the implementation of the different analyses. You can also render directly this interface in cheaper notebooks and check out quickly what code they typed. And if you're like me and your code's a little sloppy, just to crop the photo here. And also if you're in a meeting, you're talking about different data, different plots, and you're wondering, for example, hey, which samples have a segmentation? You can easily look back at the tables that you've output into the Quilt package and help you sort of filter and browse which samples may cover that application without having to install additional software or do a big setup of like bioinformatics. Yes. And also, I think Johan liked a lot to learn the inside presentation, but as you actually showed the amplicon sequencing looking at the different variation that might be in those sequences. You can also directly use IGV, which is a really commonly used tool to help you visualize different alignments and genomic data interactively. So you can do this directly with Quilt. Here we were able to visualize the AM files, which are the genomic alignment files from the SERIC pipeline outputs in an interactive way to help you visualize the mutations. So for anyone not familiar with IGV, kind of like read-based plots is this chromosome across the top. You have all the different bases of DNA and header specific location you're looking at here, the gene, and you can see all the different reads that align to those different locations in your RAM file for some example. Now in oncology, people look to look at the different mutations, genes, where you can see the near reads here are highlighted in red. So you could use this to completely check how confident the coverage of your region of interest if you're looking to profile samples, Harvard, social data. In addition to sort of interactive IGPU, there's also a really nice view I would show in a bit, a demo, where you can pre-specify different variants in the stable and just click through them, and it will quickly render the different genomic locations across different samples, showing you a snapshot of how those alignments look across the different samples related to the SERIC pipeline. So this is a really cool feature. I had sent many emails, grad school to my supervisor, telling me where the damn files are, so like screenshots of IGV, just very sloppy, very chaotic. So it's really nice to have this sort of built in right to the Quilt package alongside my SERIC outputs, so you don't need to know where the BAMs are anymore. You can just browse the interface and check it out interact. I'm going to switch over and just kind of show how this package looks like. If you know what this package looks like, So you see lots of screenshots of interface, but here's a package that we created with the static output of those 9 samples that you grab from the public CCLE database. There's different directories created for all the different output files that are generated by the static output. And we also in here have all of our input files nicely organized with this notebook. And you can add additional information like how you grab those fast cues. So if you're for people like me, you can always look back for the next time you have to do that. And you can tell really easily when you did. So additionally, how did all the static output, nice copied stuff here. You can also add like README files, so you can describe the project, helpful links. So you're get like me, and additionally we end up Quilt pipeline. That's a really nice sort of README file with all the different information, including the run command that you use, which links to all the different inputs, outputs, as well as information dropped in our little kind of similar to what Yohan shown. The static pipeline also outputs a little board, which is really handy, really big for the splitting pipeline, where you can get information right up front on the QC of the different splitting files and also their encoding. So it's really nice to have this just right here. We could interact with you, so to scroll through. Cool, check out all the different things that you need. Trace out. Additionally, as we showed, you have the interactive IGV viewer here. So you can see there's one track for all different samples. That was the static output, which is the science sequence here. And you can also sort of, grab the track and slide around. Interactively, you can also type in different gene names and grab the positions up here for the top search bar, and it will automatically sort of populate those things here. So this is really handy. You do not have to have IGV installed locally, and also not have to be able to load those damn files locally, especially if you're not a foundational biologist. It's pretty hard to email that file.
Is that a BAM file with a B? BAM. I don't want those BAM files.
And then so where's that? This is another example where you load some common input using here, and so you can sort of click through the different variants of this table. It will automatically snap into that position and put that samples where you can sort of see these variants live. So this is really helpful if you have a targeted project, if you care about genotypes, don't have to remember the locations and take it into the interactive browser. I shouldn't use the simply state at near this table and just click through different queries. Okay, and my favorite part here is sort of this way to interact with these different data that you output. So as computational biologists know, you make lots of files, and it's great to be able to shove them into this package, and then you can send these links to your collaborators who all may be looking at the same exact file and interact in the same way. So a lot of times we have questions like, which sample harbours mutation when you're working with exome data? You need to specifically search, for example, that column, ask yourself, plot all the columns that have mutations in this KRAS gene, just a file that was generated from those notebooks looked at earlier, and you easily create these samples. Very collaborators who don't know how to use the type you speak on our can also discuss. So let's create the team. In addition to being able to search your file to have clear cells the rows, you can also plot this data in interactive manner, which I can imagine means super handy for meetings and sort of reduce your workload having to make slides if you could just sort of generate these plots on the fly. So here you can also, which variety of the plots you could do, you can was on C1, I guess, would need plot the amount of mutations per sample. We regenerate these sort of lower level plots that are really informative. Best time to find each of it.
Okay.
So with that, I show you how you can use Quilt packages to help maximize the utility of your genomics data once it comes off the sequencer. So from my perspective, there are a few things that really stood out about the utility of being able to use Quilt. The first was I felt it really made data manageable, digestible, and usable across different teams. I love being able to have everything in one place. So have the input data and the output data, the exact commands run, all the logs, the reports, and also being able to visualize that data in one place, where you just have to go to kind of one location. And you can see all I thought is really handy and a great way to organize. Sending links to colleagues, like imagine me, super helpful. I worked a lot sort of across the biology team with the combo. team. They're constantly sharing files with each other, so it was really handy. As India Burn Christian did it, led itself to be able to share these domain links that links right into the versions of data we're speaking about all our meetings. And this would have been a great way to sort of minimize hiring training needs for industry, which, high consuming that could be at times. So it will be great just to be able to pull up the Quilt package and sort of leisure meeting discussion right off of there. And I think also be able to respond to follow-up questions and build back goals. And then, I think also it would help you maximize the discovery, interpretation, this sort of next steps in your data by reducing the barrier to access it across your teams. So as at least I think Edge-based is very usable and easy to interpret. I don't think you can see compile or Edge in person, so I click around on it, and then you see that your biology teams probably easily access. So to me, I guess maybe have some more